The following is a video abstract for a manuscript appearing in press online July 9, 2018 in the Proceedings of the National Academy of Science. N6-furfuryladenine is protective in Huntington's disease models by signaling Huntington phosphorylation. The first author is Laura Bowie, PhD candidate, and CIHR PhD scholar. This involves the work of several labs, including Nicholas Hertz with mitochinin in Stanford, Christopher Ross at Johns Hopkins in Baltimore, David Litchfield at Western University in Ontario, the mouse work of Simonetta Scipione at the University of Alberta, and the Ray Truant lab at McMaster. The manuscript ends with a model of N6-furfuryladenine signaling hypothesis in Huntington's disease neurodegeneration. The model starts with a trigger of DNA damage by age-onset accumulation of reactive oxygen species, or ROS. This is known via the Fenton reaction to be able to generate furfuryladenosine on adenosine residues in DNA. These modified bases need to be repaired by base excision repair to net free nucleotide furfuryladenine. We and others have shown that furfuryladenine can be salvaged by the adenosine phosphoribosal transferase and then further modified to generate a novel triphosphate referred to as kinetin triphosphate. One of the kinases that can use kinetin triphosphate is casein kinase 2, and this was the work from David Litchfield that enabled us to show that CK2 can use KTP as well as ATP and GTP on Huntington to phosphorylate it, as well as canonical CK2 substrates. The net result of the Huntington phosphorylation is participation back in base excision repair and DNA repair through the ATM complex. The role of Huntington in the ATM DNA repair complex was published by us in 2017 by Tamara Mari, and the role of Huntington as a ROS sensor at the endoplasmic reticulum moving to the nucleus was published by us in 2016 by Laura Di Giovanni. What this model does is show us a positive cascade that can be triggered by the initiation of this signaling, in that KTP signaling to produce phosphohuntingtin results in the production of more KTP by repair of the DNA. And this signal is naturally dampened when the DNA is entirely repaired and there is no more source of furfuryladenosine and hence furfuryladenine. All aspects of this model, we believe, are proximal to the DNA damage, and this is particularly well demonstrated in our cell biology data. On the top panel, in response to DNA damage by irradiation with a 405 nanometer laser, we can see the accumulation of casein kinase 2 as expected and as shown by others, but we also see the accumulation of phosphohuntingtin. Also at the area we can see accumulation of the oxidized base N6-FFA riboside in the DNA and the accumulation of the salvager APRT, and the sites of APRT accumulation match the sites of phosphohuntingtin. But we do not see phosphohuntingtin present at DNA damage if we inhibit the ATM pathway by using chemical ATM inhibitors. This gives us a proximal and temporal view of this signaling pathway, in that we believe that Huntington phosphorylation is downstream of ATM kinase activity, which is downstream of CK2 signaling as a result of nucleotide salvaging of the DNA repair product. In the context of other neurodegenerative diseases, we may have some hints as to what the trigger is of the energy crisis within the cells and what we see in Huntington's disease cells. HD has classic examples of inverted ATP-ADP ratios that we can see from models all the way out to human brains. In ataxia with oculomotor apraxia via mutant XRCC1, in a manuscript that was published in 2017 by the Keith Caldecott lab, ROS DNA damage induces parylation and hyperparylation of chains which cannot be resolved by mutant XRCC1 in that disease. But we know that hyperparylation also results in inhibition of glycolysis and inhibition of ATP export from mitochondria via AMP inhibition due to catabolism of PAR chains. In the context of Huntington's disease in the clinic, ROS-dependent DNA damage and repair and the energy crisis that results in this, we think results in protein misfolding, as ATP is essential for chaperones at the endoplasmic reticulum, where most of Huntington resides. And these pathways defining DNA damage repair and energy metabolism are all highlighted in genome-wide association studies in Huntington's disease at 6,000 and now up to 9,000 individuals, as well as relevant to other polyglutamine diseases such as the spinocerebellar ataxias. That work was published in 2015 by the Genetic Modifiers of Huntington's Disease Consortium in Cell, and covered nicely for the other diseases in a review written by Sarah Tabrizi and Leslie Jones in 2016. The Truant Lab at McMaster University is supported by operating grants from the Huntington Society of Canada, the CIHR, and the Kreml Foundation, as well as support for members of the lab from the Huntington's Disease Society of America and by Mitokinin.